Oops, that button. That button. Okay, there we are. There. Okay, so any questions from last time? We finished up metabolism. Again, the study helps on mastering are really good. I did try to explain a little bit more detail in the learning objectives, kind of some things that you're gonna want to know. So I went through glycolysis and tried to ask you questions, Krebs cycle, electron transport chain. So use that as a resource to make sure you're kind of dotting all your I's, crossing all your T's. Microbial genetics. So we're going to spend a couple lectures talking about microbial genetics. This is chapter 7 in your book. So the topics that we're going to look at, structure of DNA, hopefully that's review. We'll start with that, talk a little bit about what that looks like in prokaryotic cells. Go through replication. So that's when DNA copies itself. We call that replication. And then when DNA gets made into RNA, that's transcription. When messenger RNA is translated into protein, the language of amino acids and proteins, that's translation. And then we're also, not till next time, I'm sure, be thinking about how does the cell keep from making proteins that it doesn't really need. So there's some regulation they're going to talk about. And then what happens if the DNA sequence is altered? Maybe some ways that can happen, some ways mutations happen. We'll spend just a brief amount of time on how a cell might be able to fix those mutations, and also talk a little bit about horizontal gene transfer. So that's kind of where we're going with this. You may have some, if you've taken genetics or you've taken biochem, you might have a little bit of a leg up, but I don't assume that you've done those things. I do try to compare a little bit. We're talking mostly about genetics of bacteria. I do at times like to compare, hey, this is different in eukaryotes, what you probably studied before. All right, so start at the beginning, the structure of DNA. Hopefully you could kind of write this out a little bit. Like sometimes I have students do this before I get to this slide, say, hey, what's the structure of DNA? You know that it's made up of sugar, you know that it's made up of phosphates and nitrogenous bases. All right, so how do those all fit together? Well, this kind of diagram is what I'm looking for here. So structure of DNA, the sugar is deoxyribose. That's a pentose sugar. So there's actually, well, we'll look at that in the next slide, structure that a little bit more carefully. But that's those orange pentagons there. Of course, the greens are phosphates. That's what we call kind of the ladder of the double helix. And then the nitrogenous bases, you can see that they're bonded here with their complementary base on the other strand of DNA. DNA is always double-stranded. Always in humans, it's always double-stranded, always in bacteria. The exception there is that there are some viruses that have single-stranded DNA. Okay, so the bases here, adenine and guanine, those are the purines. I always remember that because I went to an ag school and I just remembered Aggies are pure. So adenine, guanine, Ag, pure purines. Actually, I'd say Aggies are too pure. Okay, and that means the purines have 2 rings. Okay, and then the pyrimidine bases are the thymine and the cytosine. Okay, so you're always going to, and so the A's always bind to the T's, the C's always bind to the G's. All right, so if I asked you, if I said, well, we know that the 25% of the bases in this nucleotide are thymines, Okay, you could work out how much, then you have 25% adenines, and you could work that out actually. Okay, so this is your book's graphic looking at the structure of DNA. So I'll point out a few more things about this. You don't memorize the structures of the bases up here. That might come in handy later on when we think about some mutations. The hydrogen bonding, I didn't really mention that before, but that's kind of important. When we have an adenine and thymine base pair, they have 2 hydrogen bonds. When we have a guanine and cytosine, they have 3 hydrogen bonds. So that's, if the base pairs only have 2 hydrogen bonds, they're a little bit easier to pull apart or unzip. So we'll kind of mention that later on. Of course, DNA is a double helix, and it's double stranded. So that structure was elucidated in a paper in 1953, 56, something like that, by Watson and Crick. I have that written somewhere. 1953. At that time, they didn't do any experiments to figure out the structure of DNA. They just kind of took experiments that everybody else had done, wrote a single-page paper of how they thought that all fit together, and got pretty famous for that. So you'll see that we call DNA anti-parallel. Whoops. Not sure if there was. I think I'm going to put too many R's in there. Parallel. So that means that one end, the 3' end, matches up with the 5' end. So they're going in opposite directions, basically. And let's remember what that 3' and 5' means. If we take a look right here at a deoxyribose sugar, we number those carbons. All right, so right here where there's an oxygen, you start to the right of that, and that here's the 1' carbon, the 2' carbon, here's the 3', 4', and then the 5' carbon actually sticks up. It's not part of that ring. Okay, so there's 5 carbons, we number them. So the 5' carbon here, that's the 5' end. The 3' carbon is down here, and that denotes the 3' end. Okay, and there's actually a 3' hydroxyl group that sticks down here. And that's important as we think about the directionality later. New nucleotides only fit onto that 3' hydroxyl group. So yeah, more about that. You can see the other strand is facing the opposite direction here. So we're going to number these 1, 2, 3, 4, 5. Kind of got to go around the other way because it's oriented the other way. So again, 3' and 5' end. Sometimes some of the nomenclature here can get a little bit oppressive, but down here, a nucleoside, that is the base and the sugar. And then technically a nucleotide is the base and the sugar as well as the phosphate. Okay, so nucleoside versus nucleotide. I'll try to use those terms correctly, but it gets kind of nitpicky. All right, so we touched on what the structure of a bacterial genome looks like when we were going through the structure of a cell. In terms of chromosomes, prokaryotes, bacteria in particular, they are usually going to have 1 chromosome, although there are some exceptions there, and they are usually circular. Okay, now I know in this picture that you can't really tell that. This is a transmission electron micrograph, so you're taking a cross section of the cell. So kind of what you see sticking up here, kind of just bits and pieces of this big kind of jumbled mass. Okay. that circular, when the chromosome is in a circular form, really protects the DNA better. Eukaryotes are typically going to have a linear chromosome. And they have to have some special ways to protect the ends. The ends of the chromosome are going to be susceptible to endonuclease or nuclease enzymes in the cell that would degrade the chromosomes. So when the chromosome is in a circle, it doesn't have to worry about the ends degrading. Okay, so it's a little bit more stable. Prokaryotic chromosomes are always haploid. That means there's one copy of each gene. We have diploid chromosomes. That means we have 2 copies. Okay, we have 23 pairs of chromosomes. Okay, so DNA is always double-stranded, again, for both bacteria and eukaryotes, but the bacteria only have 1 copy of it. And for bacteria, it's the chromosome is going to be found in the nucleoid region. Found in, whoops. Okay, so it's not bound by a membrane, but usually the chromosomes are all kind of found in the chromosome, that one chromosome, or if there's 2, they're going to be found together in kind of one region of the cell. Still considered the cytoplasm. So nucleoid region of the cytoplasm. And I should say eukaryotes, linear chromosomes, they are usually going to have multiple chromosomes. But remember, they do have DNA in eukaryotic cells that is like prokaryotic chromosome, like prokaryotes. The DNA of mitochondria is like bacteria. Okay, so there is a little bit of DNA in our cells that kind of looks more like bacterial DNA. Okay, bacteria, in addition to a chromosome or a couple of chromosomes, are often going to have plasmids. And again, we just kind of mentioned these briefly before. I think I showed this same diagram before. But these are really small circular molecules, DNA, that don't replicate necessarily on the same timetable as what the chromosome does. They don't encode for things that are absolutely necessary for the survival of the cell. So for instance, you're not going to find the enzymes necessary for glycolysis on a plasmid. You would find that on the chromosome. So what sort of things do plasmids, what kind of traits are they going to encode for? Here's just some examples of different kinds of plasmids that a bacteria may have or it may not have. It may have a fertility plasmid. Okay, and we've mentioned conjugation before in relation to that structure, the pili. We're going to get to that hopefully next time, a little bit more about what conjugation is exactly, but organisms can do that if they have a special kind of plasmid called a fertility plasmid. They might have a resistance plasmid that carries instructions to enable the bacteria to withstand exposure to some antibiotic. Probably one that you haven't heard of, bacteriocin plasmid. Bacteriocins are very specific molecules that can kill cells that are similar to the cell that secretes them. So maybe the same species, if they don't also have the bacteriocin plasmid. Okay, so this might sound kind of like antibiotics. Scientists kind of argue about whether to call these antibiotics. They're very specific antibiotics, if you want to call them that. So sometimes bacteria kind of work together and they form biofilms. Sometimes they have ways to kill each other, kind of whatever's in their best interest. And then kind of a general kind of plasmid, virulence plasmids. These would carry instructions that would enable an organism to be more successful in establishing some kind of infection. Okay, so there's a lot of different examples of that. Oftentimes that can confer the ability to make some kind of toxin that can make it, again, more likely to establish itself and cause an infection, say, in our bodies or in some kind of animal. All right, so thinking big picture a little bit before we get into the kind of molecular level of how DNA copies itself. But Watson and Crick didn't really, again, they didn't do any experiments. They suspected that DNA replicated itself in a particular way, but again, they didn't really know. So there was three kind of hypotheses about how DNA replicated itself at the time, and we're talking like early 50s. One way was the semi-conservative method. And if you happen to print these out in black and white, you might want to go back and kind of color code this a little bit because it won't make sense if it's all black and white. But the semi-conservative hypothesis was that one of the old DNA strands was conserved when it copied itself. So you can see here, the red strand was conserved, and then the pink strand was made from that conserved strand. Okay, so you got two new strands. That's a semi-conservative model. The other hypothesis was we had what we called the conservative method, where you can see that the old strand is unchanged and somehow a new strand was copied from the double helix was copied from that old strand. Or maybe it was by some kind of dispersive method where the DNA was chopped up and you've got, stretches of old coupled together with stretches of new. Okay, so those were the three hypotheses. How did they decide which way was the way that actually happened? Well, there was a very famous experiment published in 1958 called the Messelson-Stahl experiment. And this has been called the most beautiful experiment in biology. So I thought that we would spend a few minutes on it here, even though your book doesn't talk about this too much. It was considered so beautiful because it not only proved that one of those methods was the correct way, but it disproved the other two methods. Okay, so it's just a really nicely designed experiment. So I'll walk you through this. I'll also refer you to a YouTube video that probably talks through it a little bit more eloquently than what I will. But basically what Messelson and Stahl did, they grew some bacteria up on a heavy form of nitrogen, N15. So it had an extra neutron, I think. Because DNA is really rich in nitrogen, nitrogenous rich bases, so they grew it on heavy 19. And then after, then they switched mediums and grew it back on regular nitrogen, N14, and stopped the growth after one generation. So after one generation, they took the DNA and spun it down in a centrifuge and found they got these kind of hybrid bands. When the bacteria were grown on that heavy nitrogen and you extracted the DNA and spun it down, you got a band much lower here. So the DNA was heavier. After one generation, it was a little bit lighter. And then after another generation, you'd have to exactly know the generation time of the bacteria. They found that they had two hybrid bands, which was right here, and then a lighter band right here. And if they went on for another generation, that lighter band kind of got darker and darker. All right, so, well, so what? If you think about each of those hypotheses and what it should look like when you spin the DNA down when you do that experiment, well, you could tell right away after the first generation that the conservative method was not true, because after one generation, we weren't getting two distinct bands. We were getting one band. Okay, so we know that it couldn't, sorry, there's a little delay here. So we know that the conservative method after the first generation, we can say, well, that's not the way DNA is replicated. But this dispersive method, well, this is giving us two hybrid bands here. So we have to go out another generation to see what this would look like. Remember, if dispersive was true, we would still be getting just one hybrid band. But that's not what we saw. We were getting hybrid bands, but then when it was switched to growing on that light regular nitrogen, we were getting two lighter bands here. So it was really good evidence that, again, that the semi-conservative method was the correct way that DNA was replicated. And again, I think I pasted that website on. It's just like a 2.5-minute YouTube video that explains that. Again, the most beautiful experiment in biology. All right, so if we kind of hone in on kind of what's happening at a molecular level, the first thing to understand is that this is a polymerization reaction. So we have nucleotides that are being, is the monomer, that are adding up to one long strand of DNA. And those monomers actually aren't just nucleotides. They actually carry with them the energy to make this polymerization reaction happen. They actually carry with them 3 phosphates. Okay, so when you think about what actually comes together to make DNA, it's actually these triphosphate deoxyribonucleotides. And again, the nomenclature here gets a little bit cumbersome. If we say this correctly, for instance, say you've got a guanine base here, correctly, this would be a guanosine triphosphate deoxyribonucleotide. It's kind of a mouthful, but when you see these D-ATP, D-GTP, okay, that's the base and the sugar and phosphate, and also has with it these 3 phosphates. And we know that these are high energy bonds, so that when we break those bonds, again, that catalyzes the polymerization reaction. Okay, so this graphic from your book is pretty helpful here. You've got these, an existing DNA strand here. Remember, there's going to be a template over here on the other side. This would be a T and a G and an A base over here. And maybe, see, this is a guanosine, so must be a C here. So these triphosphate nucleotides are getting added on to this hydroxyl group here. And this is always going to be the 3' hydroxyl group. So the directionality of that polymerization is always confusing, because sometimes it's not clear if you're talking about the strand over here that's already here. So this is going to be opposite to that. Or if you're talking about the growing strand. Anyway, the new strand or the old strand. But I'll try to make that clear as we go forward. Basically, again, two of these phosphates are cleaved off. Ply2, that's even a faster, more favorable reaction than if only one phosphate was cleaved off. And now you've got, again, that guanine added on there to make the DNA strand longer. And of course, that all happens really quickly. Okay, so So where do we start? The place where we start thinking about DNA replication, we're going to start at the origin. Okay, I think that's a term that a lot of students skip over because I already know what an origin is. It's the beginning, the original, like where we start from. So the origin, that is the site on the DNA of the bacteria for the start of replication. Okay, so in this diagram from your book here, figure 7.7, again, the colors here are really subtle. They kind of look about the same, unfortunately. This little green box here, that denotes the origin. Okay, how does the cell know where the origin is? There's probably a number of signals that tell the cell where to start. We're going to talk about that. What one of them is probably a little bit of methylation there, but it's usually going to be a sequence of DNA that's rich in A's and T's, because there's only 2 hydrogen bonds that connect those, so they're a little bit easier to break apart. Okay, prokaryotes typically only have one origin. Okay, so we get 2 replication forks actually opening up at that one spot, and then all the DNA is copied. In eukaryotes, they have so much more DNA in eukaryotes that they are going to typically have many origins. So you may have seen this, you know, in the past, but here's some DNA and replication forks opening up. So there's maybe one opening here and one here and one here. Again, that's eukaryotes. But in prokaryotes, they have one origin, and then replication proceeds from both directions. So if you look at the second figure here in that circular chromosome, you can see a replication fork opening here and here, and then it's going both ways. So the light strand here is being copied from the darker purple one. And sometimes we call this a theta structure because it looks like a Greek theta right here as replication is progressing. And eventually all the DNA is copied and one chromosome goes to one daughter cell and one chromosome goes to the other daughter cell during reproduction. Okay. One enzyme that's really important to start things off is called helicase. Okay, and this enzyme, we'll say, unzips the DNA. I've more recently, instead of calling that unzipping, sometimes in the literature it's referred to as melting, so, or melts the DNA. Basically, that means the 2 strands, the hydrogen bonding is broken between the 2 strands, and the 2 strands come apart. Okay, so, and then, like I mentioned before, replication proceeds in 2 directions. All right, so your book diagrams are pretty good here for replication, so we'll follow along with those. So here's we've got a replication fork opening right here. Okay, there would be another one over here that we can't see. There's also, so here it shows the helicase that's moving ahead and actually opening up the replication fork. And there's going to be some other stabilizing proteins that just keep that double helix apart rather than it wants to kind of wind up again. Okay, and then the main enzyme involved with replication is DNA polymerase 3. And that, to me, that seems like kind of an odd name. Like, if it's so important, why is it the third one? Well, they named these enzymes sort of in the order that they discovered them. So I don't know, that's why it's DNA polymerase 3. We can see that up here. That's the enzyme that adds the triphosphate nucleotides on the 3' end of the growing strand. All right, so in other words, here's the strand that it's polymerizing. Okay, this is the 3' end here. So, right, that's going to match up 5' end here. It's going to be opposite of the old strand. All right, so sometimes you hear, oh, the enzyme moves down the old strand in the 3' to 5' direction. That's true, but it's making the new strand. It's always adding on to this 3' end because that's the end where there's that hydroxyl that's free. Okay, what else about this enzyme? It adds, astonishingly, somewhere between 500 to 1,000 nucleotides per second. Okay, so you just gotta kind of appreciate the speed of that reaction. Kind of astonishing. And even more astonishing, it has kind of a proofreading function where it can kind of look back at the nucleotide that it added and make sure that it added the right one. Okay, that matches up with the template strand here. Okay, so it's pretty astonishing that it can go that fast and still double check and see that it's added the right one. Okay, how often does it make a mistake? With that proofreading function, only about 1 in 10 billion base pairs does it make a mistake. And that's really important, that the fidelity of that process is good, because, you know, basically you're writing a new recipe card, okay? And if you're making the recipe from a recipe card that has bad instructions, every time you make the cookies, there's going to be something wrong with them, right? You need that recipe card to be good. You need the blueprint of the house to not have any errors in it, because you're going to make the house and it's going to be wrong. All right, so we need that DNA to not have many mistakes. So, but actually that RNA polymerase can't just clamp on to that DNA template and just start making new DNA from nothing, de novo. It can't actually do that. The the DNA polymerase 3 actually needs something to kind of start with, to clamp onto. It actually needs a primer. Okay, so an enzyme called primase will synthesize an RNA primer at the origin. Again, so the RNA polymerase, so the DNA polymerase 3 has a 3' hydroxyl group to start with. Okay, it can't just start from nothing. All right. It's important to note, though, that little primer sequence, this is only maybe 10 to 20 base pairs long, so it's just a little short segment. And this is going to be temporary. Okay, eventually some other enzymes are going to come through and cut that out, because you can't have a little bit of RNA stuck into your DNA strand. So eventually that gets fixed. Okay, again, it's made of RNA, so you have to excise that out. Okay, so this is what we call the leading strand, because this is the way that replication occurs. It's going towards the replication fork. Okay, so it can just go in one continuous way, because again, it's adding on to its 3' hydroxyl group, and it just can go in one continuous strand. The other strand on the bottom here, let's see, make sure, whoops, said that backwards. This is going to be the 3' end down here on the bottom. So on the bottom, it has to synthesize in this direction. All right, so we call that the lagging strand. It has to synthesize the new DNA in segments. Okay, so let's look at the synthesis of the lagging strand here. Okay. So first of all, what happens on the lagging strand, that enzyme primase is going to make a series of primers. Okay, you only need one primer on the leading strand to start out with, but on the lagging strand, you need a whole bunch of them. So that's these little things in red there, series of primers. All right, and then DNA polymerase 3 is going to synthesize some kind of pieces of DNA called Okazaki fragments. Whoops. Okay, so that's DNA about 1,000 to 2,000 base pairs long. Okay, seems pretty long, but they're going to be kind of manufactured in these short segments. And then what has to, yeah, question? Yes, yes. Okay, so let me repeat this. So DNA polymerase 3 is going to synthesize kind of short fragments of DNA called Okazaki fragments. Okay, so that was kind of those arrows that I was making on the previous slide. Okay, those are the Okazaki fragments. Okay, they're going to be, again, this is the 3' end, so this is the way that replication has to happen away from the replication fork. Okay, and then a couple of enzymes will come in and get rid of the RNA primer and fill that in with DNA. Okay, so DNA polymerase 1 will replace the primers. And does a little proofreading as well. So you can see that's right here. That's the action here. So it's getting rid of that primer there. And then that's not quite good enough to seal up all the ends. We need another enzyme, ligase, to kind of, what we say, seal the ends. I believe what has to happen there is just a few phosphates have to be added. And again, here's the action of the DNA ligase, okay, at the end there. Yeah, we have another question. Yes, exactly. In the leading strand, the DNA is synthesized toward the replication fork, and in the lagging, it has to be synthesized away from it. Yep. Okay, good. So you're going to want to, I believe that there's a nice homework exercise that has you label all these parts. So that's a good exercise. You're going to want to understand the role of all these enzymes, the RNA polymerase 3, the RNA polymerase 1, the ligase, helicase. Okay, you're going to need to know what all those enzymes do. And there is, oh, so right here, that's if you're having trouble visualizing it, this is just a short little 2-minute YouTube video that will help you visualize that. I believe there is one associated with your homework as well, but that's a nice one that kind of is right at the level of what we're looking at. Doesn't add in a bunch of things that we're not worried about. Okay. So basically that's replication. Just a couple other things to add in that are important to understand about this process. One problem that the cell encounters during replication is supercoiling. So, and you have experienced supercoiling if you've ever sat on a swing and tried to spin up. Okay, you can only spin so far because eventually, right, that chain gets down lower and lower and squishes you, right? You can't pull the chain apart any further. Here's an example with a rubber band. Okay, if you keep pulling this rubber band further and further apart, you get this kind of supercoiling back here. So eventually, you can't pull the rubber bands apart anymore. Eventually, this replication fork can't keep going this way because of the mechanical strain imposed here. So the cell has a way around that. It has an enzyme called gyrase. In bacteria, we call it something different. In eukaryotes, they're topoisomerases. But DNA gyrase, we can call DNA gyrase, I guess. In bacteria, that enzyme is a little bit different in a bacteria. And basically what it does, so we're talking about kind of upstream here of the replication fork, it will cut both strands of DNA and unwind it to relieve the supercoiling and seal those strands up again. So it's rare that one enzyme will do so many things, make the cut and seal them again. But again, this is going to relieve the supercoiling that is just part of the physics of what's going on here. Okay, and again, this enzyme is different enough than what we have in humans that we actually have some antibiotics that target that enzyme. Okay, they're fluoroquinoline antibiotics that target DNA gyrase. We'll talk about that in another month or so. All right, and just one other thing that I want to mention, because this is going to be important later when we talk about repair. There is some DNA methylation that goes on after replication takes place. So by methylation, I mean a CH3 group is added on, especially to the nucleotides that are adenine, especially to the adenine bases. So an enzyme will go through and add these methyl groups to the DNA once it's made. Why does it do that? Well, we think that it has some role in helping the cell kind of find the origin of where it should start replicating the DNA next time. We know that it's involved in DNA repair. We have enzymes that scan through up and down the DNA that's just been made and make sure that it's the same as the template strand. When it finds a mismatch, maybe it finds a G that's matched with a T, for example. How does it know which base to fix? Well, the older one is going to be more highly methylated. Okay, and we'll talk about that again later, but there is an involvement in DNA repair with methylation. And we think that maybe some DNA is just going to get transcribed more often if it's more highly methylated, so it may have some kind of role in regulation as well. Okay. All right, so let's step back again and think big picture for a minute before we move on. Okay, we like to call this the central dogma of genetics. So in other words, how does information flow? Okay, we just talked about the structure of DNA, that sequence of bases in the cell's DNA, that determines the cell's genotype. Okay, the sequence of those bases. And then how do we get from there, which is kind of like we talk about that as the blueprint of the cell. How do we get from there to observable traits of an organism? Well, that flows through messenger RNA. All right, so by the process of transcription, a messenger RNA is made from DNA. And note that when DNA copies itself, we call that replication. That doesn't necessarily have to happen first before transcription takes place. Okay, so Don't have that misconception. And also, right, when any kind of RNA is made from DNA, we call that transcription. We'll talk about other kinds of RNA, but thinking about, you know, where the information flows, messenger RNA is made from DNA by transcription, and then that messenger RNA, every 3 bases here, we call those bases a codon. Okay. Each of those codons is read at the ribosomes and matched up with its corresponding amino acid. Okay, it's transfer RNA that helps facilitate that, but that messenger RNA then is changed into the language of proteins. Okay, it's translated then at the ribosomes. Okay, and in bacteria, what is phenotype? You know, it's not blue eyes or whatever, like what it is for us. It might be, does the organism is it able to make enzymes to metabolize lactose, for example? Okay, that's an observable phenotype in bacteria. Okay, so that's the flow of information. If you get, say, a vaccine, say an mRNA vaccine, injected into your cells, there is no way that is going to change your DNA. That doesn't ever happen in our cells, right? We don't have enzymes to make DNA from RNA, all right? So note that is impossible. There are a few viruses that can do that, but coronavirus is not one of them. So again, that's the flow of information. So let's talk a little bit about RNA before we get into how it's made from DNA. Again, this should sort of be a review. It's single-stranded in all of life, except for there are viruses that have double-stranded RNA. So that was sort of mind-blowing when that was first discovered. So most RNA is single-stranded. I don't dwell on this, but just so what are primary, secondary, and tertiary structures? We think about this in conjunction with proteins as well, but the primary structure is just going to be the sequence of bases. All right, so the adenine, the thymine, the uracil, whatever. So that sequence, you know, DNA has a primary structure as well. A secondary structure is when it has kind of 3D shapes in kind of a localized area. So for proteins, that can be like an alpha helix or a beta sheet. We see a little bit of that with RNA as well. And tertiary structures just refers to a really convoluted, complex 3D structure. Okay, so oftentimes we picture RNA as just kind of being a line, but I'm going to show you some pictures that reminds us that no, this is actually, there's a lot of different bonding that takes place there, and these are actually quite complex structures, as DNA can be as well. The bases, of course, we've got uracil instead of thymine as one of your ribonucleotide bases. And then we have ribose sugar instead of deoxyribose, which is in DNA. So if you look at a structure of RNA, the only difference here is right here at this 2' carbon of the sugar. Instead of a hydrogen, like what we have in DNA, we have a hydroxyl group. So ribose instead of deoxyribose. So it's really the only difference. They're very similar structures. All right, so what kind of RNA is there in the cell? I'm going to talk about 4 different kinds. There's a couple other kinds that are mentioned in your book, that especially one's a regulatory one that we won't get into. We've already talked about RNA primers, all right? And if you think about, so those RNA primers, of course, are necessary for DNA replication. Okay, we already talked about those as well. They're really just temporary. So if you think about how much of the total RNA in the cell makes up RNA primers, like negligible, like really, really tiny amount. And then messenger RNA, that's sort of like the kind of RNA that you always think about. This is only, though, about 4% of the total RNA in a cell, because it actually degrades pretty quickly. It's produced, it's maybe translated a few times, and then it degrades, and the cell has to make more if it needs more of the protein that encodes for. This seems like a good time to bring up a couple of definitions. Messenger RNA in bacteria can be, let's say, can be polycistronic. Okay, poly meaning many, and cistron meaning gene. Okay, so in other words, it can encode for multiple genes. Okay. Or let's say, that's a little bit more clear to say encode for multiple proteins. Okay, so in other words, the information for multiple proteins is carried on that one piece of RNA. Okay, if you contrast that in eukaryotes, that messenger RNA is always going to be monocistronic. Okay, so in other words, one gene or one piece of RNA is going to give you one protein. Okay, so we'll talk more about that, because when you think about regulation and how a cell can kind of keep from making more protein than what it really needs, having more than one proteins encoded on one piece of RNA is actually pretty ingenious. If you have, say you need 3 different proteins to metabolize a sugar like lactose, it makes sense to make the same amount of all 3 of those proteins at once, just regulate them together. So it's kind of an ingenious system. Transfer RNA. We'll talk much more about messenger RNA in a few minutes. This is maybe about 10% of the total RNA in a cell. Even though they're kind of short, tiny pieces, they're only about 75 nucleotides long. Their main job is to deliver the correct amino acid to the ribosome. Okay, so the amino acid that's going to match up with that codon of messenger RNA in order to make the correct protein. All right, so if we look at, just briefly here, look at a picture of transfer RNA. So the amino acid is going to attach up here on that 3' hydroxyl group. And we're not really going to talk about too carefully how that process happens. It's a couple different enzymes will charge that tRNA, meaning add the amino acid to the appropriate tRNA. If you look at the structure here, it has some hairpin loops. And these 3 bases down at the end of this hairpin loop is referred to as the anticodon. Okay, so that's going to be complementary to the codon on the messenger RNA. And in your book usually draws tRNA to look something like this kind of bent structure. So, you know, if we were to flatten it out on paper, we would see this cloverleaf type structure, but we think what it actually looks like in nature with, you know, this true complex shape is more like this here on the right-hand side. So it always uses this kind of bent icon to denote a tRNA. All right, so more about that later when we get to translation. Most of the RNA in the cell is actually ribosomal RNA. Okay, this is about 85% of the total RNA. And it's kind of the kind of RNA that everybody forgets about, I think. But remember, this is the dominant molecule in the ribosome. Okay, so that's what the ribosome is made up of, ribosomal RNA as well as some proteins. Okay, and ribosomal RNA can be catalytic. In other words, it can act like an enzyme. So when it does, then we call it ribozyme. Okay, so again, that's RNA that acts like an enzyme. Okay, so that's going to be responsible for ribosomes, are responsible for a lot of what takes place in the ribosome, like forming peptide bonds between amino acids, for instance. So we'll talk more about that later. Just a little review of the ribosomal structure in bacteria here. We've got a small subunit and a large subunit that come together during initiation of translation and read the information on the messenger RNA and translates that into protein. Okay. All right. Oh, and I just wanted to show you this picture just to kind of appreciate the complexity here. I didn't post this, but this is the 16S, so it's one strand of ribosomal RNA in the small subunit of a particular bacteria. And again, if we kind of mapped it out, this is what it looks like. We think the structure actually is kind of all, bonded together like this. So the brown is the ribosomal RNA and the purple are proteins associated with that. So just to kind of appreciate the complexity of ribosomal RNA, not to memorize there. All right, so at least let's get started here with the events of transcription. Okay, so really important as we move forward to keep kind of replication separate in your mind and how that works with transcription, and then definitely don't goof up transcription with translation. So that's going to be your main goal, to kind of keep those processes separate in your mind. Okay, but transcription is when we make RNA from DNA. All right, and we usually describe this in kind of 3 stages. We talk about it in terms of initiation, which is, in my mind, probably the more challenging. That's when the main enzyme here, RNA polymerase, binds to the DNA and all those signals that it gets in order to bind. Okay, and then elongation, when it's actually reading the DNA template and just adding on those nucleotides, ribonucleotides. And then how does it know when to stop? That's called termination of transcription. And there's actually 2 different ways that we think that may happen in a cell. Sometimes, 50-50, so it might happen one way, it might happen the other. So we're going to go through both of those methods. Okay, so the elongation part is easy, but talking about how it starts and how it stops is a little bit more challenging. Okay, with replication, it just starts in one place and then the whole DNA is copied. It doesn't have to be like a stop to it. So it's just a little bit more complicated. All right, I think the way your book covers transcription in particular is kind of lacking in some areas. So I wanted to bring in a couple other pictures that I hope will clarify things a little bit. I want to introduce some terminology that we're going to use a little bit later. So again, hopefully that's helpful. Sometimes when the book kind of glosses over things, it kind of makes you wonder, well, how does that really happen? I don't get that. So we're just going to add a little bit more detail in some places. Okay, so again, this is a graphic from outside your book. Orient yourself here. This is the DNA down here, and of course up here, the DNA. Only one strand of the DNA gets copied in replication. Okay, either one strand or the other. I suppose there could be a time where maybe two different enzymes would be going the opposite direction, but typically either one strand or the other. And our main enzyme here is RNA polymerase. So we're going to talk about how it binds to the promoter here in a minute. In this picture, it's just showing elongation, how the RNA polymerase is moving down the DNA and making this new RNA transcript. It's adding those triphosphate ribonucleotides. I want to talk about the terminology up here on the DNA strand. So in this picture, they call this strand that is being copied, they call that the antisense strand. Okay, so in other words, this is the negative strand of DNA. Why do we call that the negative strand? Well, this messenger RNA, assuming this is, let's call it messenger RNA, that's being transcribed here, assuming that that strand gets read at the ribosome, we always call that plus sense RNA. Okay, so RNA that's readable at the ribosome, it's the plus sense. So that's going to be copied from a negative sense. All right, so this becomes important when we're thinking about viral replication. You're never going to get a plus from a plus. Okay, if you copy a plus strand, then you're going to get a negative strand. And sometimes it's helpful to maybe, let's write in some bases here to try to understand some of this terminology. Okay, I can't write as little as what they have here, but say the negative sense strand here on the DNA is CCAGT. Okay, then the complementary strands on the RNA, those are the complementary bases. All right. So this other strand of DNA that's not copied, that's also the plus. So that's going to be the plus sense. So one DNA strand is going to have a negative and a positive. In other words, this is going to be the sense strand up here. You'll notice that this sense strand of DNA, this is going to be pretty much the same as the RNA. So in other words, this would be oh, right, okay. GGTCA. Okay, all right, so you'll see that it's GG, except RNA has uracil. Okay, so how do you know which strands get copied? Basically, the cell knows. Because if this strand were to be copied instead, that's going to be a totally different messenger RNA, and it's not going to be readable at the ribosomes, most likely. Okay, so that's a little confusing. The other names that we have, this strand of DNA that's copied, we refer to that as the template strand. So if you use something as a template, it's kind of what you're using to copy. So like you might trace around a template. Okay, so that's the DNA that's copied. And then the other strand of DNA is called the coding strand. Again, template strand and the coding strand. Okay, so the coding strand, as you'll notice, looks exactly like the messenger RNA code. Okay, yes, question.
So
Yes, yes. Okay, so I know it's confusing to have so many names to call one thing, but you see it in different places called different things. Okay, so the question was, is the coding strand always the same as the sense strand? Yes. And the antisense, that's always the template strand. Okay, it's not that, oh, the bottom one's always copied. It's just one or the other. If this was being copied up here, oh, then suddenly this is the template up here. So whatever is used is the template strand. We'll go from there. Okay, so I'll try to kind of use those terms as we go. Yes, another question. So how are you supposed to know which is which? Right. So basically, say this strand right here that's being copied. Okay, that's the template. Okay, so you wouldn't know ahead of time which one it's going to be. But so knowing which one gets copied, then we can say, yep, okay, this is a template because it's the one that's being copied. Okay. Yep. And then the other strand is the coding strand because the RNA is going to look just like that. Okay. Okay, so let me just take a minute and compare this enzyme that it works in transcription with the enzyme that is the star player in replication. Okay, because RNA polymerase definitely works a little bit differently. We saw that DNA polymerase 3 needed another enzyme, helicase, to unzip or melt those two strands apart. RNA polymerase unzips the strands itself. Okay, so it does a lot of work. We saw that DNA polymerase 3 needs a primer. Okay, no primer needed. It does have to recognize certain sequences, and we'll look at that in a minute. Of course, as the bases involved in DNA and RNA has uracil instead of thymine. That should be review. And we talked about DNA polymerase 3, how it has really good proofreading, rarely makes mistakes. For the RNA polymerase 3, the proofreading is much less efficient. Okay. In fact, I think your book in one of the tables said it doesn't proofread at all. My understanding is that it's just not very good at it. So it makes errors maybe 1 out of 10,000 times. 1 out of every 10,000 base pairs are mismatched. Okay, so the fidelity is not as good, but again, as long as the blueprint is okay, the next time the RNA is made, it will probably be fine. So it seems like, still pretty good. All right, so I know we're getting to the end of our time here. Let me at least introduce how transcription begins. And again, I've taken a figure that's not in your book to try to explain this a little bit better, but I want to drive home a couple of points. First of all, orient yourself. So this is the DNA strand. And this is a sequence a little bit upstream of where transcription actually starts. So upstream is kind of to the left, right? And we call this region the promoter region. So the promoter region is where RNA polymerase binds. Okay, we think that RNA polymerase has some helps in binding. There's something called the sigma factor. So that's Greek lowercase sigma, basically the sigma factor. That's going to transiently bind to RNA polymerase, this great big enzyme, and helps it bind to this promoter region. Okay, so helps RNA polymerase bind. The more tightly RNA polymerase binds to this promoter region, the more likely it actually is to transcribe the gene. Okay, and I'll write that in a second. We think that there may be times where the cell, it doesn't need very much of whatever that gene is going to be. So if the RNA polymerase only binds a little bit, well, sometimes it gets transcribed and sometimes it doesn't. Okay, so certainly a cell could have different sigma factors that help the RNA polymerase either bind really tightly or really loosely. Okay, so let me write that. The tighter RNA polymerase binds to the promoter, the more likely it is that the gene will get transcribed. Okay, kind of floppy. Hopefully you can read that. Okay, and there's two other regions that we know are particularly important for that RNA polymerase binding. That's -10 base pairs from the start of the transcription site and -35 base pairs. So again, these are two regions that, for whatever reasons, whatever that sequence is, seems to be especially recognized by RNA polymerase. This -10 region is so important that we have a special name for it. That's called the Pribnow box. Okay, and this is only in bacteria. In eukaryotes, we call that sequence something else. We call it the Tata box, which seems to make a little bit more sense when you look at the sequence of it. But in bacteria, for whatever reason, we call that the Pribnow box. And again, that's a region that's just especially important that the RNA polymerase recognize it if it's going to bind to it and if it's going to be successful in transcribing that gene. Okay, we'll pick up there next time. Again, be working on that chapter 5 homework and get that out of the way. I hope you have a good day.